Good morning, dear students. Welcome to my new video, the cell cycle and mitosis cell division. Now, as you can see here, you can see different stages of cell cycle. As you can see here, the cell cycle is divided into 4 parts. The first part is known as G1 checkpoint, second is S phase, third is G2 checkpoint, and the fourth one is the mitotic cell division or mitosis. But dear students, there is one G0 phase in between every M, that is mitosis, and G1 checkpoint. So let's see how these phases work. The gap 1 checkpoint, the S phase, and the G2 gap checkpoint, together known as interphase. Let's just start with the G0. In this, while some cells are constantly dividing, others are quiescent. These cells exit G1 and enter a resting state called as G0. In G0, a cell is performing its function without actively preparing to divide. G0 is a permanent state for some cells, while others may restart division if they get the right signals. Okay, let's take a look at the cell cycle of a proliferating human cell. As you can see, the G1 phase time duration is approximately 11 hours, S phase 8 hours, G2 phase 4 hours, and the mitosis completes within 1 hour. So it means that it in total requires 11 plus 8 plus 4 plus 1, that is 24 hours for one mitosis completion indirectly. Now let's see what happens in G1 phase. In G1 phase, the cell size increases in size as well as the cellular components are duplicated. In S phase, DNA replication completes and each of the 46 chromosomes, that means 23 pairs, is replicated by the cell. It means there are in total 92 chromosomes in the cell's nucleus right now. Next phase is G2 phase, in which the cell grows more and the organelles with the proteins develop in preparation for the cell division. After G2 phase, M phase is there, that is the main mitosis division. Mitosis is followed by cytokinesis, the separation of cells by forming a furrow, that is also known as septum in case of bacterial cell divisions, which decides the boundary between two newly forming cells. Furthermore, there are two steps in the mitosis. First one is known as karyokinesis, in which the nuclear material, that is chromosomes, double and divide separately into two nucleus, which is followed by cytokinesis, in which after the division of nucleus, the cytoplasm separates by the formation of a cleavage furrow, separating two new daughter cells with their two new nuclei. So let's see the mitosis. The mitosis itself is divided into 4 phases: prophase, metaphase, anaphase, and telophase. So let's see what happens in prophase now. Prophase is the first stage or first phase of mitosis. During prophase, the complex of DNA and proteins contained in the nucleus, also known as chromatin, which is thread-like, fiber-like, get condensed into X-shaped chromosomes. The chromatin coils and becomes increasingly compact, resulting in the formation of visible chromosomes. Chromosomes are made up of a single piece of DNA that is tightly and highly organized. The replicated chromosomes have an X shape and are called sister chromatids because they have doubled in the previous phase, that is S phase. The sister chromatids are pairs of identical copies of DNA joined at a point called as centromere. The centromere has site of attachment for spindle fibers known as kinetochore. Then a structure called mitotic spindle begins to form. The mitotic spindle is made of long proteins called microtubules that begin forming at opposite ends of the cell. The spindle will be responsible for separating the sister chromatids into two different cells. Prophase is followed by the second phase of mitosis known as prometaphase. Prometaphase is the second phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During prometaphase, the physical barrier that encloses the nucleus, called the nuclear envelope, breaks down. The breakdown of nuclear envelope frees the sister chromatids from the nucleus, which is necessary for separating the nuclear material into two cells. Another important event during prometaphase is the development of a protein formation called kinetochore around the centromere, the central point of joining the sister chromatids. Long protein filaments called kinetochore microtubules extend from poles on either end of the cell and attach to the kinetochores. Prometaphase is followed by the third phase of mitosis known as metaphase. Actually, prometaphase and metaphase makes one phase, that is the actual metaphase. The next phase is the metaphase, that is the third phase of mitosis. The process that separates duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During metaphase, the cell's chromosomes align themselves at the center, that is equatorial plane. The chromosomes which have been replicated and remain joined at the central point, called the centromere, are called sister chromatids. Prior to metaphase, protein formations called kinetochores formed around the centromere. Long protein filaments called kinetochore microtubules, that is spindle fibers, extended from poles on either end of the cell and attached to the kinetochores. During metaphase, the kinetochore microtubules pull the sister chromatids back and forth until they align along the equator of the cell, called the equatorial plane. There is an important checkpoint in the middle of mitosis called the metaphase checkpoint, during which the cell ensures that it is ready to divide. Once the cell has established that all the chromosomes are properly aligned and the kinetochores are correctly attached, the cell enters the fourth phase of mitosis known as anaphase. Anaphase is the fourth phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical nucleus materials which are yet to be formed. Along the equator of the cell, on the equatorial plane, the sister chromatids are pairs of identical copies of DNA joined at a point called the centromere. During anaphase, each pair of chromosomes is separated into two identical independent chromosomes. The chromosomes are separated by a structure called the mitotic spindle. The mitotic spindle is made of many long proteins known as microtubules, which are attached to a chromosome at one end and to the pole of a cell at the other end. The sister chromatids are separated simultaneously at their centromeres. The telophase is the fifth and final phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Telophase begins once the replicated paired chromosomes have been separated and pulled to opposite sides or poles of the cell. During telophase, a nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. The chromosomes begin to uncoil, which makes them diffuse and less compact, along with a process called cytokinesis that divides the cytoplasm of the parental cell into two daughter cells with the help of a cleavage furrow. Now let's see the real mitosis in human cells with photographs in onion peels under the microscope. And this is the real mitosis of human cell. As you can see here, prophase